Professor Stevens from Heidelberg, and the topic is selection and orientational aggregation in population dynamics.
Thank you very much. Thanks to the organizer for the kind invitation. It's a real pleasure to be here. And in my talk, I will be going threefold, and I would like to tie in with the talk of this very first talk of this morning, especially alignment, but also with several talks we had over the last days. And I try to make the connection, yeah? So it will also be a bit about chemotaxis, but what I'm aiming for is an idea, basically, how we can deal, that's the overall central topic, how we can deal in the future, what are good models to describe cell pre-differentiation and differentiation. So I will make a suggestion there and make connections on research we have done in the group. Collaborations, this is several joint papers. Elio Espejo, who is now in Santiago de Chile, partly in Brazil. He is a former PhD student of mine, but now a postdoc. Kianka Khan is associate professor in Korea. Ivan Oprini is a postdoc in Heidelberg, and Juan Velasquez, who is here in the audience and will also give a talk. Okay. So now let me first come to the overall central topic I'm trying to aim for, and that is how could we look for suitable models for cell differentiation. I put it very general as a mathematician, but what we are aiming for are, of course, specific examples, but what would be good model systems? You could think about different things. You could think about bifurcation, stochastic bifurcation, and I will just make one suggestion, and I'm not sure if this is the right one, but I would like to stress a bit that we should search a bit more for these models. So what do you usually have if you look at cellular differentiation? And now I'm focusing in the beginning very much on a suggestion made by Cornelis Weyer, who is in Dundee and is a developmental biologist who deals with on one hand side with Dictyostelium discoideum, so the amoeba where all this Keller-Siegel model was about, and this blow-up question I shortly address again. This is basically a summary from some talks of yesterday, and also with chicken cells. Yeah. So what do you have? So he made we had a talk, and he made basically a conversation, and he basically said the following. So if you look generally in cellular systems and you look at differentiation, in his case, for instance, when he looks at Dictyostelium discoideum, so the slime mode which does this aggregation, so where the Keller-Siegel model was set up for in the beginning, and where this blow-up question arises for biological reasons, he says now if he looks at these cells, he can distinguish very early, even before the differentiation starts, which cells become what. Yeah? So what he observes is that usually these cells vary with respect to a certain cellular function. So he's focusing very much on chemotaxis. So he was looking on chemotaxis in this case, and his suggestion is, I will address later, but let me give a very simple example which is very common in developmental biology. So even if humans develop, what you have when you have this first tissue blob in the beginning, for instance, it's a standard sorting process and a differentiation process. If you have this tissue blob, the cells are seemingly the same, whatever it means being the same. There's a variability with respect to the adhesion receptors, and what you have in these first tissue blobs in the very early stages of development is that the cells with fewer adhesion receptors basically sort out to the outside. Yeah? That doesn't mean anything. You still have the tissue blob, but what you see is basically you have a spheroid of tissues. You have cells in here. They are seemingly the same. Let me mark the other ones in whiter color. So you don't see at first glance a genetic difference. You just have a variability with respect to the adhesion receptors, and then later, if you look a bit later, and you can also see this in artificial tissues. That was what we had connections with in Leipzig with the biotech center. You see the same, that you have basically the cells with fewer adhesion receptors here on the outside. Yeah. So you have a variability. You have a cell. One of them has a number of adhesion receptors. That means it sticks to other cells or sticks to the ground a bit better, and the other one has just fewer. So the same with us. We have a certain variability in height and strength and length. I don't know what. So the question and suggestion was, when you have this sorting, still the cells are seemingly the same, but once you have the sorting done, of course you can imagine, for instance, and now I'm getting a bit more vague, you have, for instance, nutrients coming in, oxygen or whatever. It's easy to imagine that these cells have a much easier life to take up the oxygen than the cells inside. So what the suggestion by Weyer is not necessarily with this problem, but with, I come back to that later, with the chemotaxis in Dictyostelium discoideum, when you have the cell sorting, which you also have there in a more complicated fashion, that such a variability gives rise to a structuring process. Here it's really an ordered process, and that then basically this ordering process can result in differentiation. So, for instance, suggestion, these white cells really become finally different from the other cells. Yeah? So, suggestion, and I come back to the real problem of Kesvaya later, but this is a very common problem I also like to address in the future. And there's, to be honest, there's a nice stochastic model now by Voss-Böhme and co-authors, but it's purely stochastic on a cellular automaton on cell sorting, but it's rigorous in the sense, in the stochastic setting, that they prove that there is a phase separation. Yeah? This is 2010. There's no limit. I'm still thinking about the limit. It's a purely stochastic model, but the result is rigorous on the stochastic particle level. Okay, so let's look at this, and the question is, what are good models to describe this? Of course, I could always say, okay, I have one cell type, I do a model for that. I have two cell types, I do a model for that. But what is our task as mathematicians in the life sciences to describe this switch from 1 to 2? We have seen that already when we look for the models for the evolutionary traits and so on. Yeah? So that you basically try that the system selects. So here you could say there's a variability, but then the system finally selects for a fixed finite number of sets. Okay, so question of this talk, and then I will combine a lot of things, and I hope you can follow. This is all the thoughts in my head. I hope you can still see the red thread. Okay, the question is variability in cellular function the basis for pre-differentiation? Let me call, I don't know if this is a general biological term, probably not, but let's call that pre-differentiation for the moment. And then you have a structure formation process, which is obvious biologically in this setting. And can this lead to differentiation, and can you describe that? What are good models to do that? I just will make one suggestion. It doesn't mean anything. I just suggest to think about that more deeply in the future. Okay, so now let me come to the system which was actually addressed by Kesvaya, and since he's dealing with Dictyostelium discoideum, I come a bit back also to this chemotaxis issue, because that's the species which is one of the model organisms for chemotaxis. And there you also have, in the later stages, once you have the aggregation, you have also a kind of pre-differentiation and differentiation process and a sorting process. Yeah? Okay, so we have seen that already in talks. We were more talking about bacteria, but the questions of blow-up came up in the early times, and they are of high biological relevance in the Keller-Siegel model for this species, Dictyostelium discoideum. So you know already from previous talks probably that under starvation conditions, these amoeba produce a chemical attractor, and they sense the signal and move towards higher concentrations. So now a short detour on chemotaxis. Not all species produce the chemoattractant themselves they are reacting to. So you have a lot of things. You have pattern formation, self-organization, and all these kinds of things. And depending on what you have, that means also that depends on which question you address. I mean, if you address, if you want to see a pattern like in E. coli, if you want to model self-organization, your second equation, the reaction equation, will be different, yeah, and also your chemotactic sensitivities, and also the question if you want pattern and global solution, or if you really want blow-up for biological reasons, is also different, yeah. Okay, but chemotaxis is very common, also in our body. We have seen pictures also by Martin Burger and so on. So it's very important, and also in our development, as a human development, it's very important in the early processes. You have a lot of rearrangements of cells and cell death and growth. So you have, this is a very interesting process among several, and is very important for spatial reorganization. So now let's go back to the questions of Cornelis Weyer. Should put that here. So he's in Dundee. So when you have the aggregation of Dictyostelium, which many people dealt with these questions on blow-up and chemotaxis, you later have a mount formation. And what you have, and I show a picture after this, you have that the cells start what, let me call that pre-differentiation. I'm not a biologist, just to say I don't want to fix this term. But what you have is something like here. So you have cells which have a certain variability. The question is, what is the variability? And basically in the experiment, Kesvaya can predict before something like the stalk and the mount is, the stalk and the fruiting body is formed, I'll show it to you in a minute, which cells will be where, which is funny. You don't see the structure, the final structure, but you can already distinguish for certain reasons when you analyze that very carefully experimentally, and he is doing that among others, which cells will be where, in average, not a single cell, but on average. And what the claim was, and that is also handled by several people by now, or also in former times, like Kevin Painter, for instance, in this mount, these cells vary with respect to their chemotactic sensitivity, and his question is basically, can this variability result in the cell sorting, and then the structure, and then in final differentiation. So it's another example for this type of thing. So now let me show the picture. Many of you may know. So, okay, in this, chemotaxis models are very general, and also the Keller-Siegel model is very general. So it's a very general model where you basically describe the cells, and where at those times when the model was set up, basically people said, okay, you have something, and that was variable, could depend on the cell density and on the chemoattractor. Let me call it V. And then you have this term where you basically move up the gradient, but also there can be variability, the so-called chemotaxis sensitivity, which can also depend on U and V and grad V. So it was, when it was set up, a very general model, and I will stress this shortly just to make it clear that if you're asking for global solutions or blow-up, it has very different and very important biological reasons, and especially also the blow-up. So and then you have a reaction kinetics, and that depends really what species you have. Do you produce the chemoattractant? Don't you produce it? So first of all, it may diffuse, but the question is, do you produce it? Which is not always the case. Sometimes it's just a chemical which comes from other species, so you would not have this production term here. Let me call it just a general F of UV. And I mean, also depending on what you are modeling, you have to look here what you are doing, basically. Okay, so and what Kesvaya can say, you see this mount here. And what happens here is what I stressed before, is this cellular sorting. So what you're after is basically, this is the final stage. The aim is somehow that the spores, that the cells survive. In the beginning they are all the same, they are all separated, and you have this self-organization process, and always, this is a long-standing model problem, so many of you may know, so it's a bit of a repetition. But what is interesting that the species seemingly completely individual cells, which do not have a difference, they don't know about each other, you can put them together, and they are able to build something which looks like a superstructure. And the funny thing here is that it can even build something which looks like a worm and move. This is a separate question, but it's really interesting. So you can separate them, really that's what the biologists do, and they still can do this. Okay, and that was the thing which was first addressed with this now so-called classical Keller-Siegel model, was to understand this. I will very shortly address it, and then I'm after this here, and especially after the cell sorting. And then the big question is, what are good models for cell differentiation? But that will just be a suggestion. Okay, you have this, you want to survive, there's a starvation condition, so the aim to do this structure is very relevant biologically. You try to have 10% of the population survive, the rest will build the structure and die. That's the reason to do this all. And then they can wait and overcome starvation conditions. Okay, what is Kesvaya doing? So we are just here, the classical chemotaxis is maybe here, and we are looking for the self-organization process. So I'm not looking at pattern formation, which is also the case like we heard in E. coli and so on. Then of course you want global solutions and patterns and so on. So you take these terms, as we have seen in several talks yesterday, also more general, and that's what's set up by Keller and Siegel in this general setting. But if you look at this species, you want the blow-up, and it's crucial to, and this I will explain. So that's biologically crucial to have the blow-up. Okay, now we are looking at this case, and Kesvaya says, okay, he finds, if he looks at variability, he finds cells which are not so effective in chemotaxis on the top of the mount, and cells which are more effective in chemotaxis on the top of the mount, and cells which are less effective and not so fast, not so direct in their movement on the bottom. So funny thing is, you would say, okay, these guys are fast in chemotaxis, they are the good guys, they will survive. No, on the contrast, these guys will later form the stalks and die, and these slow guys will be the ones which are in here. And the funny thing is that he can cut this thing, separate it, take the upper half, and again the upper half, if it's not too few cells, can again build such a structure, and the lower half too. Yeah? So again, these cells will, although they are weak or strong, again can separate. And what he can do is he can check basically how fast they are. He can label cells, and then basically decide already in average on this stage where these cells will finally end up on average. Yeah? So it's a very interesting, and that addresses again, I hope I could make this clear, this question very early, their fate is on average determined, so that means very early you could, and I could, as a mathematician, basically decide what's going on. Yeah? There's no surprise, there's no brain, let's say. The cells is a structure from a process where the own dynamics basically construct the structure. This is more complicated. There's a lot of differentiation going, lots of cell types, so I stop here. Okay, now let's shortly come to this blow-up issue again. This is just a repetition, so I will not do that very long, just to make it clear. This is what I just explained, so I can skip this. Yeah, that's what I explained. You want to understand if this chemotaxis can be a cell sorting issue, and his question is, he asked basically, can chemotaxis be the main mechanism for the sorting process in this mount? And this we try to address. We are not yet completely successful. There are also cellular automaton models which address it, but we are tackling this question. Okay, question now we try to address. Does chemotaxis serve as a main mechanism for cell sorting in the mount stage of DD? So a bit of repetition. We know all that there was this question around, which is very old. Also Childress, before Keller-Siegel, the model was around, addressed this. Question at that time was, is chemotaxis in this species, who is self-organizing, which is self-organizing, the main mechanism for self-organization? This was answered, and the main and first basic paper on this, Childress already gave a hint. Yeah? So he, for instance, gave already the very clear hint that in 1D there's no blow-up for chemotaxis. It's a very old result. He had also suggestions for the 2D and the 3D situation. There were some little errors in it. And then Jaeger and Lucas basically addressed this question in 2D. And let me just say why this was so relevant biologically to look for the blow-up. There's a bit of a misconception in the literature. You have seen the picture, so you saw this. You basically have cells on the agar. So it's first a two-dimensional, spatially two-dimensional phenomenon. So now you want to say, if you want to prove, which is done, that chemotaxis is the main mechanism for self-organization, and you look at a spatially two-dimensional problem which describes the two-dimensional motion, and you see that this process is self-organization, so it's a 3D final outcome, the 2D model has to have a blow-up in finite time, otherwise you did not catch the main mechanism. Yeah? So it's of high relevance if you want to prove that mathematically that you have the blow-up in the 2D model. And that is what Jaeger and Lucas basically found out. The answer was basically yes, also mathematically, if you have the right situation in the general chemotaxis model, you can have global solution and you can have blow-up. And they said basically you can have both. That means, and this is also happening, if you have two weak cells, you don't get the self-organization. And they showed basically that it depends very much on how high the production of the cells is, what the initial mass is, and that fits very much to the general biological setting. Yeah? And that was the reason, because there's a bit of a misconception in the literature by now, that this was of utmost importance, just to show, of course the biologists knew already, that you need the blow-up to prove biologically that chemotaxis is can be the main mechanism. Yeah? So that was a biological answer. So in that sense, the blow-up was the biological answer, but you are looking for the self-organization. If you look for pattern formation, it's different. Yeah. So just to make there's not a kind of fight between blow-up or not blow-up. It is in some species of relevance, in others not. Okay, just that for the summary. Okay. So now let's come to the sorting. Yeah. So I have to say, if I want to do a model for the sorting, I would really be three-dimensional, need to be three-dimensional. We are not doing that yet. That's my aim with the group. And you would do something, you have to look at how the particles move in this mount. Yeah? And that is there are some pictures also, among others, from Kiesweyer. You have so you have this mount. I mean, the blow-up model stops before. It just says, okay, self-organization can start, principally. But now we are already in 3D. So that means basically I would need to write down a 3D model somehow, for instance, take a variability with respect to chemotactic sensitivity, and then say somehow I get a kind of sorting. Okay, we still need to look at a good model for that. This is open, but we did something which I would like to call a test problem. So let me explain that to you. That's a bit further away from what we want to do, but let me explain why I had the hope to get a hint with this test problem. Yeah? And finally, it's a very nice mathematical problem. What we said, okay, let's go back, because now we don't have all the paths, how the passes of the cells are. We have to, they also crawl over each other, so there are some biomechanical forces involved. I don't want to do a completely big model. I want to have a basic mechanism, so we are still thinking about that. Yeah. But let's try to get a hint, if we could get a hint for the question of Case-Weyr, if you can have something like that, that you have, let's say, two cell types. Of course, you have variability, but let's assume you have a very weak chemotactic cell type and you have a very strong chemotactic cell type, which is already a simplification. Let's stay in two dimensions. That means, of course, I'm not at all in this situation. Yeah? I'm basically in the situation like before, where people try to show the self-organization mathematically, which was, of course, known biologically much longer. And let's see the following. If maybe we are lucky, yeah, this is philosophy now. This is not what we finally want to do, but let's get a bit of philosophy. Let's look at the stage before in 2D, and let's see, maybe we are lucky and you can distinguish the types already beforehand. And then you can say, okay, let's see if one type blows up, let's say this guy, in a time earlier than the guy who is weaker. In this case, let me stress it again, the blow-up has not the meaning of sorting, yeah? Just to be sure. What I want with the blow-up is basically that the self-organization strength of this type is in time earlier than of this type, and then still you need to do a 3D model to see if there's a sorting. But if that would have been the case, I would have been very hopeful that we take the effort to go into this model. Yeah? So I hope you understood me, otherwise ask me later. So this is simply a philosophical test problem, and I want to see if I can distinguish the types like the biologists can, mathematically with respect to this behavior, maybe with respect to another behavior. So we basically look, again, 2D, we look at two types, and the hope was that one type blows up at a time earlier than the other ones. Both self-organized, so both should blow up, and that was a test, yeah? So now the real modeling issue is this one. Okay, let's look at the simplified model. Now this will be more mathematical. I try to explain to you why I was interested in this. So we were looking at two species, U1, U2, let U2 be the weaker one. And let's stay spatially two-dimensional. That means I'm really before this. I'm not addressing this problem. And let's see if we can distinguish them. One possibility to distinguish them is that one is doing self-organization earlier than the other one. So why did I think that this would happen? Finally, it turned out it's not the case. I thought the following. So you have the two types. You have the very simplified Keller-Siegel model, where you know that in one species situation, you can produce blow-up in 2D and also global solutions depending on these two parameters, the diffusion and the chemotactic sensitivity. And let this be the weaker guy. So then in this case, because we look at Dictyostelium discoideum, we need production because this is what they do. They produce the chemical, others don't do it. And there is a decay, so the loss out of the system. So now I thought, okay, if I look at this system and this is weak, and I would say, okay, these guys don't do chemotaxis at all, this equation is basically decoupled. Yeah? It basically acts for itself, and it will just have an influence on the production for this species, but this is positive, that it will just enhance blow-up for this species. And this will, of course, not blow up, but it doesn't do chemotaxis. So the species should be the same. They should be actually very close. Yeah? So it's not two different species. It's basically a weaker cell type and a stronger cell type. So you cannot, how to say, for this question, you shouldn't start here to saturate this equation. I know mathematically how to get this separation of blow-up and global solutions. You can do a lot of math there. But for this question, this is not the issue. The equation should structurally stay very, very close for this issue. Yeah? Otherwise, you address a different question, which is also fine, but it's different from what I want here. Okay, so now I thought, okay, if the chi2 is 0, the system is decoupled, and you can, of course, always construct, as you know, from all the papers, which I do not mention any longer, starting Jaeger-Lokhaus, and then all these papers which were mentioned during the recent talks. You can do a lot of things about this equation. And then now let's switch on an epsilon of chemotaxis. And my hope was I can have this just blowing up, a very weak blow up, a very late blow up. And if I just put an epsilon here, this still may blow up later or not or whatever. And the equation for itself wouldn't blow up. Yeah? If you have here, you know that from the 2D problem, if chi2 is very small and diffusion is very large and you forget about species 1 and you have a low production, this would not blow up because you have exactly the switch in 2D. Yeah? You can have both situations. Okay, so another question was if this is a very weak blow up type and this is an epsilon, so for itself a non-blow up type, can I separate the blow up? This was my hope. This is not the case. We were quite surprised. That's part of the PhD thesis of Elios Perro. And what did we get? So we did it radial symmetric. So now Suzuki, for instance, analyzing the general spatial case and Elio also. Let's look at this classical chemotaxis. You know that from the talks yesterday and before. Let's look at this case and let's take the elliptic situation, and you can always reduce it as it was given in the paper by Jaeger and Lucas. You can always non-dimensionalize that you basically end up with these parameters. You just have 3 parameters here. We are in the bounded domain, non-boundary condition, the radial symmetric case. So all these things of classical solutions, if the parameters are right, were proven in Elios' thesis. Also Wolanski has a very general systems on multi-species chemotaxis, also with more species here in the chemical. So and Dirk Hausmann is just having a preprint and others on existence results of solutions. But this system also can show blow up. Okay. So we looked at that. So if you consider the radial symmetric situation, you rewrite the system in terms of the rescaled mass function. Then you can rewrite the equation for the chemical like this. This is classical and was done very early already in the papers by Juan Velázquez, Miguel Herrero. And then you get equation for these rescaled mass functions. We have boundary conditions, and the goal was, can we find sufficient conditions for blow up, and if possible, can we prevent, prevent is to my taste the wrong word, does the other solution still have global solution, because we want the blow up at least for one species, or for the other one too, but later, yeah? So that's not the issue to prevent something, but this is biologically makes sense to have the blow up here. Okay? So the result is the following, and that fits very well to the case for one species. So if the M1 is smaller than this term, or the M2 is smaller than this, and let's try to interpret at least this one, that means this mass is basically smaller than 1 over chi. 1 is the chemotactic sensitivity. So if the chemotactic sensitivity is large, this term is small. That means you need a very low mass to have global solution. That's classical. And if the chi1 is small, this term will be large, and then you can still have a large mass and not get blow up, yeah, because the chemotaxis sensitivity is too weak to create the blow up. That's classical also in the situation we know from here. Yeah, that fits to what we know from one species. So and then blow up, generally we don't know which, or if both or one species blow up, so your one should also always be the stronger one, happens if this is fulfilled. And that basically is the similar, now you have switched the sign, yeah? And that means basically when the chemotactic sensitivity is small, this term is large. That means the mass, if you have a weak chemotactic sensitivity, you need a lot of mass to have the blow up if you have it at all. And if the chemotactic sensitivity is strong, you don't need that much mass. The system is still driven to the self-organization. And this is a classical condition that you concentrate around 0. We have pushed the blow-up point basically into the 0 in this Petri dish. Yeah, that's the setup of the model. Okay, that fits.
Sorry,
yeah. Yeah.
Silly question. I don't pay attention. Is 2 equal to 8 pi?
The last condition does this correspond to the?
This must correspond basically to the classical thing. So maybe but I have to check. Yeah, could be. I mean, this fits to the classical condition, basically, yeah? So it's probably the pi, yeah, sorry for this, yeah. Thank you. But I checked, but probably you're right. Basically, this is all fitting to the classical one case. So what we showed is basically, if U1 blows up in finite time, then also U2 blows up at the same time. So bad luck. Yeah? So what I had hoped is not right. So the species are not distinguishable in this model. Yeah? At this early stage, that was what I had hoped for. So that was a wrong hypothesis, but it's a nice math, so I was still very happy. But the result is not what I had hoped for. So that means basically, but then now, since I know the result by now, we can think differently. What you have basically, if you have two species here, the conditions for the U1 are for the blow-up. The U2 itself is not fit for a blow-up itself, but since the V is driven with the U to the steepness, which is sufficient for blow-up, it just catches the U2 and takes it with it, although U2 itself would never blow up with this condition. Yeah, it couldn't make it itself, it's just taken along with it. The hope that the blow-up time is later also wasn't correct. Okay, whatever. So I was a bit disappointed. I was very happy about the math because it's a nice result. And but the biology is sort of different. Okay, the proving methods are classical. We stick very much to the results by Herrero-Velázquez. So you assume that the U1 blows up in finite time and the U2 is bounded, and then you construct a lower bound for M1 and an upper bound for M2, analogously to what you have done here. And then you construct sub and super solution, and then with the comparison argument get the contradiction. So the difficult thing is to construct the right sub and super solutions. So the second thing, which is up to now formal, is the following. Okay, I was a bit reluctant then to go immediately to the 3D problem. I wanted to see a hint. Yeah, I tried to explain that philosophically, but who knows? There's a lot of things going on. It's a 3D motion. You can follow the path of the cells. So Kisvaya can label them. You can basically follow several cells and do the path analysis. But okay, that's for the future. But then we said, okay, there must be a difference. Okay, this is so far formal. A derivation of the blow-up profile. So you can calculate the asymptotics for these parameters. You can also do that rigorously. We have not yet done that. And you take specific parameters for this one formal calculation for which the M2 of the weaker species is much smaller than the M1 near the blow-up point. Then you do the asymptotics, and we have the singularity. Yeah, we have conditions for a singularity, and then you see U2 is also having the singularity, but the amount of mass is much lower than the amount of mass of U, which is captured in this blow-up point. Yeah? Okay, but that's not a surprise, and you can vary the mass. So that is the difference between the species in this specific model. Okay, now I had this red thread, which probably even I have not really forgotten about. But what does this have to do with what I wanted to tell you? Namely, the main aim of this talk was to look for models for cell differentiation. This is a good problem. You have the sorting, there would be differentiation, but I was basically after something else. I was after the following question: what are good models to describe the specification? I mean, from one cell type to two or more. So let's come to this. Yeah, what we had said is basically when you have variability with the cellular function, in this case, which is of course not solved by now, so there was just one result. We also have this variability. We don't have two types, as I said in the beginning. That was an oversimplification. We have type which are strong in chemotactic sensitivity, and there are weaker ones. So if you are a biologist, also Kisvaya says he can at least clearly distinguish 5 types, but I mean, of course, he's sure there's not 5 types, there's a variability, and the cells are genetically seemingly the same. So they just vary, like when we run to the Wolfson court for lunch, or we don't. Some are faster, some are slower, so it's the same here. But basically, you would say they are still all the same. You know? They are still the dictyostelium cells. But so what is the question? You have variability with respect to cellular function, and Kisvaya's suggestion in this context is, again, variability can result, it's a kind of pre-differentiation, because this variability results in a sorting process, in a spatial sorting process, and this sorting finally results in differentiation. Yeah? So let's combine this. So what does it mean? If the cell differentiation directly relates to the cellular function, in this case, it would be chemotactic sensitivity and a variability with respect to chemotactic sensitivity. In this case, it would be, if we accept this biological hypothesis, would be variability with respect to adhesion receptors and then sorting with respect to the number of adhesion receptors. So if the cell differentiation you finally have directly relates to the cellular function, then what would be a good model? It's just a suggestion. Yeah? You can, as I said, you can think about stochastic bifurcation. That's not my main field. So I thought differently. The model dynamics should select. You have stages, so you have here an additional parameter like chemotactic sensitivity in the function. Here you have an additional parameter like number of adhesion receptors or density of adhesion receptors. So you have a function in space and time and a cellular stage parameter. Yeah? So the cellular stage would here be the number of adhesion receptors, and the cellular stage would be here the chemotactic sensitivity, if it's fixed, let's say constant. So and then if this is good as a good model for cell differentiation, my suggestion would be, but this is just a suggestion, that then you write down the dynamics of this model depending on what you are modeling, and you have the stage parameter, and then the model should select in its own dynamics from the continuum of stages, which describes the variability, to a finite number of stages. Yeah. So and then we are back to equations we know. This is just a suggestion. This I would call pre-differentiation, and then all this spatial sorting and stuff, which is not in this suggestion yet, would happen. So to look at this question, we basically took a model we were looking for very long time, which is a structural model also in bacteria, but this is with this aim in mind. Yeah? So let me go twofold now, if you admit. We looked very often in such alignment problems we were addressing this morning. And of course, this is a structural problem, has at first glance and second glance nothing to do with what I want to say here. But we have an alignment problem, we have orientation. Let's call that for the moment, to have this philosophy in mind, let's call that for the moment the stage of the cells. It's the orientation, yeah, because I'm looking for the basic concept of equations. And then we have a continuum of stages, and in this bacteria I was looking for a long time. You have a selection in the dynamics for a finite number of directions, here it's 2. And if I just take the equations without thinking about the bacteria, I could say, okay, I have a problem where the dynamics select for a finite number of stages. Yeah? So now let's look at this problem. For the moment I'm fine, completely fine if you just look at this structural problem, but I try to convey that I have more in mind, not with especially this model, but what I'm trying to suggest is that these kind of, not necessarily only kinetic equations, but these type of equations, and probably adding some other things, could be a good model concept to describe cell differentiation. I hope I could convey that, yeah? And that's it. That was somehow try to make you look for that a bit, first of all myself, look a bit more for that in detail. Okay, now let's look at these bacteria, and if this is hard to, if I couldn't make myself understandable in this respect, just look at the structural problem, and now we are in an alignment problem, yeah, for the math. Okay, we have looked at bacteria, we have seen the flocking, and in this bacteria what you have is basically that they are rod-shaped, so one should also look at other models which are known for rod-shaped polymers, but we looked at a specific model. So you have elongated bacteria, the stages are the orientations, and what they do, they align like the birds, but they don't care if it's head and back. So what you finally see in the experiment is that you have an axis of nearly one-dimensional motion. Yeah? In the selection, it's the last time probably I addressed the selection issue. For the selection issue, if this is a correct model, as a basic type, as a basic mathematical type, you would have the selection of all these orientations. The system selects for 2 stages, for 2 directions. Yeah? So then the orientation would be the cell stage parameter, and now forget this and we go to the alignment problem. Okay, we took a model which was suggested by Alt and Geigand, and also Mogilna and Lavinchak. So that is Alt. And it's already very old. And of course we need to compare. We have seen a lot of alignment problems, and I think there's lots of things going on. Now Lavinchak, I have to check how she's written. I will give that to you after the talk. So that's classical. For the moment, I don't put in spatial dependencies, which is of course important, yeah? Because for the maths we are doing, we still look for additional techniques, basically. So now the question was, we also asked, like speakers before, we want an alignment, and we want to define as possible as general kernels for alignment as possible, and classify the geometry of the kernels to see alignment or not. So it should be, you have a structure, you want to count the number of orientations you finally have, and you want to say what geometry relates to which orientation. Yeah? Okay, so we take no spatial measurement for the moment. You can add that, it's clear how to add that, but let's look at this problem because we want to see something analytically. We wanted to classify these kernels. So you have basically, this is the cell density in time and in orientation gamma, you are on S1. So this is normalized just to minus 1/2, 1/2. So you have, at the moment, the general turning kernel that you say you want to reorient. This depends somehow on F. Let's be as general as possible. Of course, we cannot be completely general. You want to align from gamma, you want to move from gamma to orientation gamma prime. And here, these are the guys who move from gamma to gamma prime. So now this is the specificity of the turning. And what is general will be this V. Yeah, so what we finally want to understand is the structure, which structures of this V result in alignment and in where is the direction you finally align to. It's not so important for this biological situation in this case, but also how many orientations do you get? 1, 2, 3, 4, 5. Okay, the other ones are things are more specific. So what you do is basically, this is a Gaussian. If this is 0, you have a Dirac, yeah? And this will be basically, this measures the accuracy of reorientation. These are the guys, this is the guys who are here, basically, the guys who move from gamma to gamma prime. And this is what you see, basically, if you interact with the partners in between. And the V will define how you interact, how sharp this is. So you basically move here, you want to move here. And your final, these are the interaction partners, and your final situation could be in the half, but that's not defined. This depends on the V. And then the sigma basically measures how sharp, how precise this is. Yeah? So the sigma would measure, do you do a precise alignment, let's say in the middle for the moment, for the intuition, or do you do an unprecise one? That means you will be somewhere in this cone with higher probability, for instance, in the middle. Yeah? And that basically would describe like what we had. Basically, when you say, you can also say that the precising would be gamma prime equals gamma plus V of W minus gamma. And the interaction partners are these W's. Yeah? These are all the guys you interact with. And now how you draw the V says basically what you can do. You can be symmetric, asymmetric. You can attract, repel. This is all described by the V. Yeah? So now what we want to do in an ideal world, to say nothing about the V and define all geometries of the V and then derive from the structure what is the internal function. Of course, we can do it fully. So let me tell you what we can do. And that G is the standard periodic Gaussian. Okay, and let me give the results and then. So with this bacteria, you have something which you would call bidirectional alignment. This is a specific setting, but we are looking a bit for the general one. Let me call it attraction and repulsion. You don't have a head and a tail, so if the angle is small, you try to attract, and if the angle is larger, you attract to the end of the other cell. And what was done by Geiger is, for instance, if you take the precise turning, let's assume for the moment for the intuition that you turn into the middle, then you would take the sigma equal to 0, you have a Dirac mass, and you have something like deterministic turnings. We have a very precise alignment, which is of course not natural. Then convergence of solution could be proved of G sigma to the solutions of the model with the G sigma to the solutions of the Dirac mass. And now we wanted to look at the patterns. First thing we did was we could show for the model as I wrote it before that there is local stability for alignment in two opposite directions. That means if you have initial conditions which are already having two directions, this is locally stable, but there is no selection of mass. So I took the model I wrote down first, and that is all we could get. That's joint work with Kang, Patam, and Velasquez. I will not address that because then we changed the model. And that is in the next paper. We had a closer look at the case where sigma is larger than 0. That means we have not a precise turning. You have basically a variability to move somewhere in here with higher probability to the central point. So if the orientational angle V is very small, you can approximate this kinetic equation by the following equation. This is the total mass, and this x relates to the gamma. Yeah, it's just a rescaling, so I just have a different name. This relates to the gamma. We constructed the steady states, And an equivalent formulation for these is the following. You can basically steady state this is 0, you integrate once, and let's take the total mass equal to 1. Yeah? Then you have the following integral differential equation. And then let's look what this is doing. So we could construct the steady states and judge, depending on the geometry of the V, if there was a selection, if there was a selection of two orientations or one or more. But let me give the heuristics for the end of the talk. So it's completely rigorous, and now let me give just the heuristics for the outcome. We were looking for the selection. We want finally the two orientations, and this should depend on the geometry of the V. Yeah? So if the sigma is 0, that's heuristics now, but it's all proven, then the equation I derived reduces basically to this integral differential equation, integral equation. And then you can quickly check that any function which is a Dirac in two direction, let's say you are on S1, 0, and the other point, let's call it -1/2, so you scale, and the opposite point, so there's a difference between 180 degree, then any function with different masses, or same masses, can fulfill that. That was what we had already with this local stability result. We had local stability, but we didn't have any selection of mass. For this bacteria, of course, it's important. They don't care where they are moving in this case. Yeah, this is not the flocking of birds. They just have equal mass distribution. Yeah? Or if you think back about the selection issue, if you say finally these cell types should basically become prominent, you want because usually in biology you want basically not nearly a fixed percentage, but a very precise percentage of cells becoming one type and a very precise percentage of cells becoming the other type. So you want to be precise in the selection of mass. Yeah? Okay, so that in the full model, in the original model, that doesn't work. But if you have this uncertainty added, so that you're not precise in turning, then you can get the selection. Okay, let me quickly give the heuristics for that. Now suppose you are in an F sigma case, and you have such and this converges, it's all heuristics at the moment to give you the argument, and you have already these opposite points, yeah? And you have masses which you don't define. And these should converge, as we had seen in Geiger's case, to the sigma 0 case. So what happens then? I want to see the selection of the mass. So if the sigma is very small, then you can approximate this F by this equation, where the V is now this combination of this turning part, which we want to specify geometrically in these two opposite points. I mean, this you can solve explicitly. You get this formula, and then you can see immediately that you get a selection of mass. Why? So let's define this phi is here, is basically the integral over V from 0 to x. You have the symmetry you had already in the V. Now let's look at the solution of this. That's all heuristics. If we assume that the phi 1/2 is not equal to 0, then if you want to have two peaks of this formula we had before, yeah, so if you want to have two peaks in here, and you look basically here, then what you need is the following. Then this is only the case if the peaks should be at 0 and 1/2. That was the construction of the deltas. That alpha phi plus beta phi x minus 1/2 reaches its minimum there. And this is only possible if you fulfill this equation. And that means basically that alpha equals beta equals 1/2. Yeah? So that's the heuristics for something we have done rigorously. But this gives you a quick hint why it works. So in this case, as soon as you have this uncertainty in the turning, but it's familiar for you in different situations. So if you add this diffusive part, which describes the rescaling of the gamma, then you get this selection of mass, which you don't have in the original kinetic model. Yeah? So the second thing we wanted to have is what is the geometry of the V if we want to have selection of one type, of one orientation, selection of two or more. How does the geometry of the V define the number of traits, or here the number of orientations? Yeah? Okay, the condition for 1 or 2 is the following. Suppose again this was successful to put in the sigma, and there exists a one peak-like solution for the F, which concentrates at 0. That's our starting condition. Yeah, we look for one peak. Then this solves this equation. This was this reformulation and converges to the Dirac at 0 for sigma equal going to 0. Again, you can solve this. It's heuristics again, but it's also shown in the paper rigorously. Then you can approximate this function by the solution of this equation. There's again an explicit formula for this. And if you now look here again in this exponential expression, then if you want, like we had as an initial assumption, you want the concentration around 0, that means you need this expression to be larger than 0. Yeah? And if you have this smaller than 0, then the peak is concentrated as plus minus 1/2, which is a contradiction to our initial assumption. Okay, this is heuristics, but what it says is these are more or less, the rigorous part is pretty simple, the conditions on V, the geometric conditions, or this integral condition is the condition on V to have 1 peak or 2. Yeah? And the rigorous thing looks pretty much the same. But here you get the insight, so you can give conditions on V. We want it to be as general as we could at least to distinguish, because we are still looking for selection. We wanted to have 2 or 3 or whatever types, and that depends on the geometry on V. Here it's orientation. Okay, that is that part, and then let me, without any I just give you a short hint that will no longer fit into this talk. First summarize and then give a hint on what else we did. But this will just be one more slide apart from this. So what do we have? Let me summarize. We have one example where variability of cells with respect to cellular function. Okay, here's the orientation. Yeah, it's a bit biologically not perfect, but it's a stage parameter. So just be abstract and say this gamma could be a stage parameter. And we have dynamics in this case where the system selects for a finite number of stages from the continuum. So that could be the trace. We have seen other models of this type. And the suggestion was to analyze not necessarily this equation, but equations with stage parameters, time, space, and stage, as models for cell differentiation, apart from other possible things like you could look for stochastic bifurcation, where you could check the percentage of which goes on one bifurcation branch and the percentage of cells which go on the other bifurcation branch. And then the percentage has to be quite defined. That's another possibility, but just not my main mathematical field, and there will be more possibilities. So that was the suggestion and the main red thread. And then one more slide, if this is okay. We also looked at this type of model with Kang and Velasquez. This is related, but not completely directly related, but has a connection to reinforced random walks. And I mentioned we had looked for that for a long time, and it's also used for haptotaxis. These kind of models are also used for haptotaxis in tumor growth. We had looked for a long time on models like this, which are chemotaxis-like, and where you have a specific chemotactic sensitivity, so the W is like the V, and you have just an ordinary differential equation, so you have just a track. Yeah? You don't have a diffusive structure, so the signal is not diffusing. You have more or less something like trails. You could also think about arms, but let's look at this mathematically for the moment. This model in chemotaxis, which is otherwise pretty similar to the classical Keller-Siegel model, it has a specific chemotaxis sensitivity which plays a role and which results from this normalization in random walks. And this part is non-diffusive. And that changes the structure of this chemotaxis equation strongly. And we analyze that now to a large extent and look basically what are the structures of this function here on the right-hand side, which distinguish between pattern formation and blow-up, and try to classify this. And that's all I want to say, because people also look for that. It's just an outlook for discussions these last days, so it's not really no longer belonging to the talk. But I just want to mention that we have a lot of progress now on this type of equations, also in several space dimensions, and this system behaves it's more hyperbolic and behaves very different from the classical Keller-Siegel model, just for people who are in chemotaxis and may like to discuss. But I stop here, and thank you very much for your attention.
Thank you very much for the nice talk. It's time for questions.
Concerning the selection problem, you had done the problem with alignment, and for the alignment one can very well imagine kind of a mechanical process, how the cells influence each other to get aligned.
Sure.
But then when you go back to dictyostelium, chemotactic sensitivity maybe has to do with the number of receptors on the surface or so on.
Yeah.
So the question is, can you imagine the process there where cells influence each other? So exchange of receptors or something like that?
Yeah, okay, this is, I mean, I think we understand that this is different and it would be a stage parameter and would not be alignment, no? We agree, yeah. And what Kiswaya thinks is, of course, that what you have is, what is having different chemotactic sensitivities. That means basically you are not effective in this directed motion, and that is depending on how you diffusion and chemotactic motion depends, of course, on how you can react biochemically, and that depends, of course, how much force you can basically have in contact with other cells or with the extracellular matrix, which is also there. And then the idea is, of course, that there are these receptors, again, adhesion receptors, and you have something similar like if you have too few receptors, you cannot move. If you have an average number, you can very effectively move. If you have too many, you are very sticky. So that relates very much. Is this addressing?
Yeah, no, but what I mean, what you would need is that you can change your, say, your number of receptors, right? If for a you want to have the
yeah, here in the chemotaxis issue, you may not because you want this interaction, yeah.
Yeah.
What you have is, of course, an indirect interaction in this case, which would be not the direct interaction, but with a fewer number of receptors, you would be not because you also have the forces between the cells play a role, yeah. And that means the one who are stickier will sort out different. It will be more like this problem, if I get your question right, yeah. But then it would be not as direct as I had it in the alignment model. It would be indirect via the stage parameter of receptors to the it would be a spatial process, and it would be indirectly coupled to the spatial process. That would be more extended. Yeah. It's not a direct that you throw off your receptor. This will not be the case here. Where you have the direct interaction probably is in the hematopoietic stem cell systems, yeah, where you really change your developmental. I mean, as people like say, is now that there's a plasticity, that you change basically the stage of your pre-differentiation stage, and that goes back and forth. That would be more direct. Here it would be indirect over the space, if I. Yeah.
Yes. I just wanted to go back to the spore formation. You said there could be a mechanism which correlates somehow the variability of cells, the ones which move faster and slower, in order to have then an ensemble where they form spores. That seems quite interesting somehow, because it means if the variability is sort of odd or changes by whatever, then all of a sudden they can't form spores anymore? Or
yeah.
is that
I mean,
absurd?
there's a mixture. So what they do is in the experiment, I have to check one thing. I have to check, but what they do so far is that they always cut this. Also Kiswaya and others cut this, and that means you take always the faster ones and the slower ones, and at a certain point, the slower ones, if you do that very often, can no longer build it. But what I'm not sure about is also because it depends on the total mass. If they can introduce enough weak ones, I have to check with them. But that is basically the intellectual biological hypothesis, that if you have two weak ones, it no longer works. Yeah? But they can still separate. If they can still do it, of course, you wonder, the structure gets also different on the long run. That's what you were asking? No, they do this kind of experiments. I just have to check if they introduce enough cells again, but they are pretty much on this. I mean, I say it, so it may be not his wording, but on an engineering type of analysis in the experiment. That's a very nice thing in this group.
It's essential to have a certain profile of variability within the cells.
Yeah, that's the suggestion he has. That, I mean, it's a claim, it's a biological claim, that's not my claim. Basically, that variability is important to have differentiation and structure. That's the claim at the moment, and he's looking also now for chicken development in a similar, but let's see. So.
Seems not to be the case, so thank you very much again.